Okay, so I'd like to introduce Fred. Fred is a postdoc at SAMS, so working on bioinformatics and genetics and everything else. So he will explain. I can't see if a slide is that now active on the screen. I want to keep looking behind me to look to the thing, but I can't see. Yeah, that's okay. Okay, thank you. I'll hand over to Fred. Thank you.
All right, nice to see you all. So I'm happy that Dave introduced it quite a fair bit already. So my talk is going to be about how CCAP envisions to deal with tsunami of data coming in. This is mainly like genetic or genomic data, but why not include metabolites at one point? So yeah, this thing we are starting to call the CCAP Bioinformatics Gateway. And it's all about making all this incredible amount of data more easily accessible to the community, to customers, researchers, people in schools or whatever. All right, so as a first introduction slide, I show you this graph, which is basically, it's possibly difficult to read, but it's the increase in sequence data over time. So it starts at the early 90s up till today, and we see like a steep increase of like nucleotide data associated to CCAP strains. We expect this to keep on increasing. Sorry for this typo there, but there is a kind of an issue, like where do you find this data? It's deposited in all kinds of different repositories and databases. So you have NCBI, is an American thing. You have ENA, which is the European alternative. You have a Japanese one. There's all kinds. There's also specific ones for proteins, so PDB or UniProt, you name it. Like all data is available, but it's all hidden in various databases. So the idea of the CCAP Bioinformatics Gateway is to kind of collect it and put it in a centralized area. And at least for CCAP strains, people can just access them and it becomes more easy. So we used to collect all this data quite manually in the past, and we put them in like long Excel sheets, and it was a lot of manual efforts to kind of collate all this information. But the idea of a project that I was involved in was, can we move away from Excel and make it a bit more automated and have a centralized and dynamic infrastructure, as I may call it like this, to store, analyze, and share all this sequencing data with the community. So this infographic kind of summarizes the big idea, which is there's all kind of different data types out there, but can we link them back to the CCAP strain cultures? This is quite a challenge because they're all in different forms. They're coming from different data sources. Different researchers around the planet have collected them. So are we. And the quality of the data often varies. So there's kind of a lot of thinking involved of how can we centralize this, tidy it up, and make it readily available. All right, so now I need a bit of your attention. So maybe it's good, and it's a bit small, so maybe move a bit closer to the screen so I can talk about the code. No, I'm just joking. This is just to say that a lot is like automated. So we need to, just by building the codes, we can just make it reproducible. We can automate the whole process. And basically all the rest of what I'm showing is built around code line. So a bit more of a global overview rather than boring you with the details is this slide. So it kind of summarizes the infrastructure in a way. So to the left, I guess, there is a thing called Toolbox, which is a computational unit we have at SAMS, which is a Linux system, where all the coding is kind of run. And this is there to, for example, collect all the information routinely from various data sources and screen, link it back to the CCAP culture, and then it's pushed down through a lot of different pipelines depending on the data and kind of analyzed. And the outputs of this analysis is basically pushed onto MySQL server, which is a database. So CCAP staff, which is this computer image that I put on, like can interact with any of the components. So they can maintain the scripts, they can run the scripts, change the MySQL database in the future, etc. And then the idea is, with the first implementation at least, is the website, which I'm going to show you in a moment, which goes and reads from this database and is based on R Shiny for those who are interested. So this is kind of the core idea. Things get downloaded on a Linux server, get analyzed, processed, pushed onto a database, and then there is a website reading the database. So all right, so how can you access this website resource that we have implemented? Just I'm gonna try and do this live for you. It's gonna make it more easy. So this is the, I don't know if you can see this. This is the CCAP website, familiar to, I guess, a lot of you. If you scroll down a little bit, there is this third box here saying Bioinformatics Gateway. So you can click on it and then it opens this web portal. So this is for now mainly focused on small or single gene data, which is the most abundant at this time. So it gives you a bit of statistics. There's a menu at the top. I'm not going to be able to show you much of this website, but feel free to click around. And I think later today there's an option to. But anyway, just to showcase something, if I type here nanochlorhydropsis, then it gives me a bit of results back. So you can straight away download FASTA sequences which are associated to it or collect metadata associated to it. But if I scroll a bit more down, you get a list of all nanochlorhydropsis strains in our collection and whether they have sequencing data associated to it. So if I click on the Oculata type strain, for example, you get a bit of information. So when it was isolated by Droop in the 50s. Scroll a bit down, and this gives you a phylogenetic tree based on 18S, and it positions it along other CCAP strains. So the one in dark red is the one I just clicked on. So you can see the biodiversity in our collection, and you might get inspired of which strains you want to order. So there's a lot of information on this page. I'm going to kind of skip through very quickly, but there's which genes and accession numbers which are available, where we detect an 18S, if that's of your interest, a taxonomy of this specific sequence, which is predicted by PR2, a timeline, what happened with the strain since it was isolated on the left in the 50s, and then apparently around 2010 onwards, there's sequencing available, there's sequencing data coming available, and then a bit of literature stuff, etc. But because I have very little time, I'm gonna leave it here, but feel free to click around on the web page, and currently is there to help people access data associated to the specific strain of your interest. Back to the PowerPoint. All right. Oh, so other things on this website is global phylogeny based on 16S again, where we basically put all the strains into a phylogenetic tree, which helps us at CCAP to kind of curate taxonomy of it. But it's also interesting for the community to kind of show them the real biodiversity we're having in-house. Another thing which is possibly what I'm kind of most proud of is the ability to blast against our database. So if you have an organism which you love working with and you have a sequence of it, you can basically put that in here and use the BLAST algorithm to kind of predict which is the close relative strain or set of organisms we have in our culture collection. So you can then maybe order them in or use them in your research as you want. This is a little case study that I want to show to make it a bit more, show the power of the tool we have implemented so far. There is a scientific reports paper a couple of years old now, which was a collaboration between SAMS and CCAP, where they screened a whole host of strains for their metabolic profile. So what kind of lipids they produce or how much lipids they produce, carbohydrates, etc. So what I did is I went to this paper, downloaded the supplementary material, which is the metabolic data, let's say. And alongside of it, I collected all the 18S sequences using my website, collected them down, built a little phylogenetic tree, which you can see in the center of the graph. And then I put the compositional data, if I can call it this way, on top, like a heat map. And you can kind of see that there is a dark area to the right of the graph with a dark purple outer layer. And this is eustigmatophytes. So it's Nanochloropsis, Microchloropsis, and that lot, which we kind of know are very lipid-producing organisms, but that's why it appears a bit darker on the screen. Anyway, this is kind of interesting for me because it puts the biotechy type of data together with evolutionary data being the phylogeny, and we can maybe infer some kind of more evolutionary patterns if we combine genetic data and compositional data. This might look scary for you, but that's why we started implementing a lot of these techniques as a kind of a set of tutorials. These are also freely accessible. I'm going to link them back on the gateway themselves. But it's basically, they contain information of how to use the gateway, how to taxonomically identify your organisms or your sequences, how to build phylogenetic trees, and how to visualize data, and how to combine trees and other data types. So all these tutorials are available, and we are planning to make more in the future. So as I said, currently it's mainly around single gene data because that's the most abundant. But in the future, as Dave and Matt kind of pointed to, data is coming pour in. So we need to be prepared, and that's, I think, the long-term vision of this project is, can we integrate genomic data on transcriptomics, microbiomes, etc. And what I think is very exciting too, but it's kind of, is outside of my comfort zone at least, is can we include metabolite data, and can we basically integrate the whole thing and make a kind of a package where clients of CCAP or the community can basically home in and see a bit of a full picture and to support research or education. So this, of course, is continuous expansion of the ways we interact or make interactive visualizations, but also give or make more tutorials and perhaps give training on how to do all this. And ultimately, I really want to encourage anyone who clicks on the website and uses it at any state to kind of feedback to us, because it's important that we make it user-friendly and we kind of make it so that you can all enjoy the easy access of all this data. That's what it's really about. So I want to say thank you to everyone at CCAP. Ceci, who is sadly not here, David Green, and especially also Christine Campbell, because they were instrumental in supporting me to engage with this, what I believe is quite an ambitious endeavor. So thank you very much. And if you have any questions, please come and find me, and I'm happy to give a bit more detail on the code or whatever. Thank you.